Hello, I'm Chris Kruger, Senior Manager for Content with the Davis-Phinney Foundation, and I'm here today with Dr. Mark Richardson to talk about gene therapy in Parkinson's and a new clinical trial that has recently launched called Regenerate PD. Before we get started, Dr. Richardson, first of all, thanks for being here. Thanks for taking the time out of your day to help our audience understand gene therapy and this trial. Before we get started, can you maybe share a little bit about your background, maybe how you came to be involved in working with people with Parkinson's, and maybe also how you came to be involved and what your involvement is in the Regenerate PD study?
Yeah, thank you, Chris, and thanks to Davis-Phinney Foundation for sponsoring the content, hosting this. So I've been involved with patients with Parkinson's disease and Parkinson's disease research since medical school. Actually switched into PhD programs so I could work on a cell transplantation project. And then in residency, had the opportunity to move to gene therapy. And so now I'm a functional neurosurgeon, which means half of my practice is deep brain stimulation for movement disorders, including Parkinson's disease and dystonia and essential tremor. And I have a lab that studies brain recordings from patients with Parkinson's disease from DBS devices. And we've been very active in gene therapy clinical trials since residency and continuing now at Massachusetts General Hospital, where I direct the functional neurosurgery program.
Maybe we can start there with a big picture overview of gene therapy. What is it, broadly speaking? Why do researchers think that gene therapy can help people living with neurological disorders in particular? And maybe it'd be helpful to show a visual aid just to give a picture of what this might look like for people who might benefit from that. And maybe you can walk us through that. So we'll put that on the screen, that image, and maybe you can just talk us through what this screenshot is showing us.
Yeah, sure. So if first you start with what's happening in Parkinson's disease, which is unfortunately cells are dying. And at some point, hopefully researchers will figure out for each individual patient what's making the cells die. And if that's a problem that can be fixed in the cell machinery, there may be a gene therapy that can do that. Another strategy is to try to rescue the cells, to try to prevent them from dying, even if the mechanism that's causing the cell death isn't fully understood. So that's the concept behind this gene therapy trial that we'll talk about. And what it does is it packages the genetic material, the DNA, for a molecule called GDNF, glial-derived neurotrophic factor. The keywords there are neurotrophic factor, which means a factor that supports neurons. So the neurons are the cells that are degenerating in Parkinson's disease. And the idea here is to give cells a lot of extra copies of the information that's required to make this neurotrophic factor, which is then secreted with the hopes that that will prevent cell death, to slow it down, and potentially even help neurons to regenerate some of the arms of their axons that reach out to communicate between brain regions and within brain regions. So the genetic material for the GDNF is like a letter, and that is packaged in the vector, a viral vector, and there are a lot of different types of viral vectors. The one that's used in this trial is an adeno-associated viral vector, and it does not replicate once it goes into a cell. So this isn't something that someone who gets this gene therapy has no chance of spreading it to someone else. It's not going to move all over the body. It's essentially going to stay in the cells that receive the package. And that package is the genetic material in the viral vector, which is like the envelope. And there's a word that's not on your schematic, which is the capsid. And the capsid is really kind of like the box that this envelope with the letter inside might be transported in. Mm-hmm. The kind of fuel behind this delivery is a direct injection. And in this case, it's a direct injection into the brain. It would be nice if you could take a pill or you could do an intravenous infusion and have the package be delivered exactly to the cells in the brain where you want the therapeutic molecule to be expressed. but the technology isn't there yet to bypass an important part of the highway through the vascular system, which is called the blood-brain barrier. So that's a very tightly controlled interface. And if you put these vectors in the bloodstream, they will go to a lot of body parts, but not so much to the brain. The good news is that once they're in the brain, this package finds neurons very well, and they then deliver the letter. Package is open, delivers the letter into the cell. The cell then can open this envelope and read the letter. And the reading is the process of seeing the genetic material. The cell uses some machinery and it makes the therapeutic molecule, which in this case is GDNF.
Okay, so let me see if I can run this down to make sure that I understand it as we go forward, and maybe this will help some folks in the audience too. Uh, as I understand it, we're saying Parkinson's is caused by damage, or one of the things that happens in Parkinson's is damage to cells, particularly neurons, particularly dopamine neurons, and there's other things involved too, maybe, but right now we're just talking about that. And so one thing that can cause that would be some genetic function that is not working properly or optimally in the body. And so what gene therapy tries to do is sort of use a sort of not neutralize, but a virus that can't replicate to inject new material into a cell that's already in the person's body and then correct sort of that with the new material, correct the functioning of that cell.
Yes, in this case, it's not necessarily correcting a genetic problem. That is a hope for gene therapies of the future.
Okay.
This one is giving extra doses of a growth factor that's going to try to slow down the disease process and prevent cell death. So, in this case, cells in an area of the brain called the putamen, which receives the projections from an area where cells die the most, which is the substantia nigra. In this recipient area, the cells are going to see a lot of GDNF. They're going to secrete that, and then the projections of dying cells, and some that are not dying yet, are going to take up the GDNF, and they're hopefully going to be much healthier and resistant to whatever the insult process is. And that could be genetic, like you mentioned. It could be some, toxic exposure of, unknown origin in the past, and other, combinations of genetic or environmental insults. And as you know, and as your viewers will know, no one's Parkinson's disease is the same as someone else's. There are really a variety of causes to what we call idiopathic Parkinson's disease.
So I think as I understand what I missed and one of the main thing I missed in my description was that this is not to correct a genetic dysfunction, so to speak. This particular Regenerate PD gene therapy intervention is designed to increase something we think is very positive and good for the brain. We're trying to produce more of that, and that's the GDNF that you described.
Correct. That's exactly right. And so this is a strategy that potentially can help all comers. So you might have Parkinson's disease that derives from a different, mechanism than one of your friends, but the therapy could benefit both.
Great. So that's important because you might hear gene therapy and think, oh, I don't have a genetic mutation, so I'm not eligible for this. So it wouldn't be relevant to me, but that's not the case.
Correct. That's a great point, because a lot of diseases for which people talk about gene therapy, the goal is, there's one cause, we know what it is, or maybe there's more than one, but we know exactly what it is, and we're going to go try to repair that thing or give more of that thing that's not working right. But Parkinson's disease is different. There are some genetic causes where there are people working on developing a gene therapy that does fix the specific problem. But as a bucket, there's a lot of stuff going on in Parkinson's disease. Sure. And so this is a therapy that can potentially apply to all those.
Yeah, so let's talk about the trial more specifically now, if we can. And I think you've already addressed some of this next question I was going to ask, but as I understand it, the Regenerate PD study is investigating something called AB1005. Can you say what that is, and I guess just clarify again, just to make sure we understand exactly what AB 1005 is maybe going to be, why we think this might help?
Yeah. So this is the trade name for well, I guess maybe it's not the trade name. It's clinical trial name for AAV2 GDNF. So this is the sponsor of this trial is a company called AskBio, and that's what the AB is the AskBio part, and this is the name for their molecule, which is AAV2 GDNF.
And so the purpose is to increase the GDNF production in the brain, and we think that will help by helping protect the neurons that are being impacted in Parkinson's.
That's absolutely right. And the term that we use that people may have heard who are watching this is overexpress. So there are many, 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 many copies of the gene. It's taken up by the cells, and they use their own machinery, just like they would translate the message of DNA all the time that's intrinsic to the cell. They're also going to find these copies that have come in from the outside, and they're going to translate the information. They're going to make GDNF, and GDNF is a molecule that gets secreted from the cell, and then it's taken up by cells that would be affected by the Parkinson's disease process.
Okay, so let's jump back to that. Now that we've talked about AB 1005, maybe we can jump back to that image, and can you just lay out for me what is AB 1005 in that image?
The DNA for GDNF is the letter. The envelope is the AAV2, so that's the viral vector. That stands for adeno-associated virus type 2. There are many different what's called serotypes. The AAV2 is the one that's been used in humans for gene therapy the most and has a really good safety profile. So that's packaged in something called a capsid, and that's AB 105.
So the combination is what the
Yeah.
candidate, so to speak, is.
It's the AB 1005.
Great. So this is a phase 2 trial, is that correct?
Correct.
And so how does this differ from the previous trial in phase 1? Can you talk a little bit about what was learned there and what's coming and why phase 2 resulted from that?
Right. So the way that we hope that the effects of GDNF overexpression are manifest in patients is to improve motor symptoms. So this is put in part of the circuit that's affected by Parkinson's disease with a specific goal to target the motor territory of the circuit, because motor symptoms are obviously would affect patients a great deal. They're relatively easy to measure, and improving these can make big improvements in quality of life, similar to what deep brain stimulation can do once people get to a stage where they have a lot of motor fluctuations. So in the first trial, for which there's a publication from 2025, there were two groups, a lower dose and a higher dose. In the lower dose group, symptoms were essentially stabilized over 18 months, which is remarkable in a group of patients where otherwise you would expect them to continue to decline. In the higher dose group, motor symptoms improved, including the UPDRS score, which is a rating of motor symptoms, and something called off time, which is the amount of time totaled in a day where someone with Parkinson's disease does not feel like their symptoms are well managed. Now, the caveat to that study was that there was no placebo arm, so all the patients received treatment.
So, I think we'll talk about maybe the eligibility and who can participate in this trial in a minute. But before we get there, can you just describe a little bit how this trial works? Maybe step by step. I assume there's a screening process and maybe some assessments. Can you just describe if I wanted to say, hey, I'd like to enroll in this, what would I be expecting to do?
Sure. The screening is done over 3 to 4 visits. There are assessments in both the off and on medication state. Includes the UPDRS I mentioned, completing a Parkinson's disease motor diary. There's an eligibility review panel that will determine if someone can move into what's called baseline period. The baseline period is 2 separate visits. It's typically at least 3 to 6 weeks apart with similar assessments as done in the screening visits. There's a PET imaging study that's done. And if the person successfully moves through those steps, which include getting a brain MRI, making sure the trajectories look safe, then they would move on to surgery.
Okay, and so you've already said that some people will be in a control group that'll have to get a placebo. So can you say a little bit about how that works? What is the surgery? What does the surgery entail? And if I am in the placebo group, would I have a surgery too? I guess I would have to, otherwise I would know.
You would have a very small surgery. So this is the way I explain to the patients that come to the MGH. The trial's appropriate for people who would otherwise be ready for deep brain stimulation. Many people don't realize when they're ready for that, but it's essentially when the medications aren't working well enough and they're getting motor fluctuations. People need to have responded to dopamine replacement therapy. And because these same people who enroll in the trial are candidates for deep brain stimulation, which we know will work to improve motor symptoms, it's very important that people understand that when they enter the trial, they may get randomized to placebo. Now, the chance is greater that you would be randomized to treatment. It's a 2-to-1 treatment to placebo randomization. But we don't want people to enroll in the trial who, whether they've been putting off DBS or didn't know about or been waiting for DBS and they really need relief of their motor symptoms, like yesterday.
Mhm.
They're not probably the best person to enter the trial. But for someone who either never wants a device in the brain or is willing to wait or really wants to have a shot at gene therapy, then it's appropriate. And then what we tell them is, you're going to come in for a surgery. It's typically an overnight stay. We have the same surgical protocol that a lot of centers, including ours, will use for a sleep DBS surgery, where there is either an MRI scanner has been converted to an operating room or an operating room has an MRI scanner in it. They're asleep under general anesthesia. The whole procedure, we say, may take 10 hours, okay, because of the setup and the time to infuse the medication. It sounds like a long time, and so people think, well, that must be very complicated, but it's actually pretty straightforward.
I want to jump in and offer a little observation here. I want to go back to something you said for a few moments ago, which was a couple things that people might think about participating in a study, or reasons people might participate in a study. As somebody who lives with Parkinson's myself, I think there's another reason that I think is worth noting, which is that one can learn a lot, even if one does get a placebo assignment in a group, for example, just because you get to spend more time with expert providers, and you can gain some information that you might not have had access to before. You get some really great time with even the nursing staff that you can connect with is really valuable from my experience of being in trials. So I wanted to share that, but I also wanted to note that one concern I would have if I was if I was considering this trial would be the question about open label extensions and whether this might be available to me down the line if I was in the control group. And is that a part of this study? Is there an open label extension possible? Might that happen?
Yes, that might happen. That is a part of this trial. The caveat is the availability of the study drug to people who had been randomized to placebo wouldn't happen until the end of the evaluation period for the last patient who was enrolled. So for someone very early in the trial, that could literally be years, depending on how long it takes the trial to enroll. For someone at the very end, that might look different. It might be more like the order of 2 years, as short as that. So that's the caveat. It's certainly the goal of the study sponsor is to provide the drug, if it looks like it's safe and efficacious, as soon as possible to people who are randomized to placebo. The reality is that could be some time for the individual person.
Can you maybe give a high level picture of what are some of the eligibility criteria on a high level, and where can we find more of them, and maybe also where can we find the study sites? I think we can put the links in the description for this, but maybe if you can just give an overview of that might be helpful for folks in the audience.
Sounds good. Yeah. So this information is available on the clinicaltrials.gov site, which is the official registration site for clinical trials in the United States. Big picture, people need to be between the ages of 45 and 75, diagnosed with Parkinson's for not less than 4 years, not more than 10, with essentially eligibility criteria for DBS, which is presence of motor fluctuation with significant hours of time in the off state, typically stable Parkinson's medication, so you can't be still fooling around with the medications for at least 4 weeks prior to the screening. So to be eligible for screening, you need to have been on stable medication for at least a month. Demonstrated responsiveness to levodopa. Exclusion criteria are prior brain surgery for obvious reasons. That includes DBS. If people have dementia or psychosis or serious mood disorder, that's a disqualifier. And then a known history or current evidence, genetic, medical, or otherwise, that their Parkinson's disease or Parkinsonism may not be idiopathic Parkinson's disease. So that's a very important one. There can't be any question that this is something else, because GDNF may not have the same effect if it's not idiopathic Parkinson's disease.
Okay, yeah, thanks for that overview. And we'll put the link to the clinical trial site in the description of this video. I wonder if you can say maybe what's the next step somebody should take from your perspective? What would you recommend somebody do if they're interested in learning more?
Well, first, you're going to talk to whoever your Parkinson's doctor is, and get their input, let them know. And you may not have ever talked about surgery before. They may not have brought up deep brain stimulation. They probably won't know what gene therapy is. Many, even neurologists are not going to be familiar with the trial or the fact that this is a really safe surgery to do. Still, I think if you surveyed most neurologists, they would be completely wrong on what they think the complication rate is for even DBS surgery and complications. So I would say, don't be discouraged if you think you're a candidate for this gene therapy or other surgeries. You can look on the clinicaltrials.gov site and you can see which site's closest to you. There are emails you can email. There's probably contact on the Ask Bio site, which is a study sponsor. The short answer is, yeah, let your neurologist know, but you may have to do your own research and own outreach for this too.
Yeah, I would say for me, when I participated in trials, I always reach out to close people with whom I'm very close. My sister is one of them, my parents, some of my close friends, and say, does this sound like it makes sense to you that I would do this? And then we can talk about it from that position. I like to bring in other people I consider part of my care team too. And I definitely appreciate your observation that talking to your own physician, that's a necessity from my perspective.
That's a necessity. And even if it turns out you're not a candidate, it highlights for them the fact that, okay, this disease is really having an effect on me. And I really, sometimes this is, I won't say lost, but, what I've seen just from my own experience with people with Parkinson's, it's like anything. No one wants to say that they're not doing great. So often you go in, doctor says, how you doing? And you want to be doing well. And so you say, I'm doing great. I'm really fine. I'm doing great. And the family member may look, oh, really? I'm not so sure about, okay, well, and sometimes you are doing great, and that's good, that the medical therapy is working, but certainly interest in this kind of thing, it helps everyone's kind of awareness and education on this.
Well, thanks, Dr. Richardson, for taking the time to walk me through this trial and the details of what gene therapy is and how it will work, how the Regenerate PD trial will work. And I hope to connect with you in the future. Maybe we can talk again as the trial reports come out, and we can sort of have a debrief about this and discuss the results of the trial when they are available.
I would love to do that. It's been my pleasure, and I hope this is helpful for your viewers.